Thanks to the organizers for the kind invitation to speak. So in the wake of the excitement following the cure of the Berlin patient, two recent transplant cases reported by Tim Henrich and the Mississippi baby case reported by Debbie Prasad have given us a dramatic indication of what we're up against in the effort to cure HIV infection. In the transplant cases, two patients received allogeneic hematopoietic stem cell transplants for malignancy from donors who were CCR5 wild type. And antiretroviral therapy was continued throughout the transplant period to prevent the donor cells from becoming infected. And after a couple of years, when HIV could no longer be detected, treatment was stopped. And the rebound, which typically occurs 2 weeks after interruption, did not occur, and instead the patients had a prolonged antiretroviral-free remission, in one case lasting 8 months before a sudden and dramatic rebound in viremia. And similarly, in the case of the Mississippi baby who was born to an infected mother and had a viral load of 20,000 on day 1 and was started immediately on an aggressive heart regimen, viral load became undetectable. Treatment was stopped against medical advice at 15 months, but there was no rebound, and this patient went for over 2 years off of therapy before there was again a sudden and dramatic rebound. And what's interesting in these cases is not so much that the rebound occurred, but what they tell us about how the virus is persisting. These patients all lacked a detectable HIV-specific immune response due either to the transplant or early therapy. And in the absence of immune responses and antiretroviral therapy, viral replication is exponential. And so persistence in these patients can only be explained by latent infection. It's not consistent with low-level ongoing viral replication or long-lived productively infected cells. Now, the only latent reservoir that's been clearly established is the latent reservoir in resting memory CD4 cells, which we originally hypothesized could arise really as a result of the normal physiology of CD4 cells, which respond to antigen by becoming activated, generating effector cells, some of which survive and go back to a profoundly quiescent state as long-lived memory cells, allowing future responses to the same antigen. In HIV infection, the virus replicates preferentially in the activated cells and kills them, doesn't replicate well in resting cells. But occasionally, some of these activated cells can become infected, we hypothesized, as they are transitioning back to a resting, non-permissive state. And this gives a stably integrated provirus in long-lived memory T cells, and with a future activation of the cells, virus production can resume. So I'd like to first describe the quantitative viral outgrowth assay that was used to establish the presence of these cells because it's relevant to several aspects of the talk. So in this assay, purified resting CD4 cells are plated in limiting dilution and then subject to activation with a mitogen that activates 100% of the cells, and cells with latent virus can then begin to produce virus particles, which can be expanded by the addition of lymphoblasts from normal donors so that you can grow out from a single latently infected cell enough virus to detect by P24 antigen ELISA after about 2 weeks. With this assay, the frequency of latently infected cells was shown to be very low, about 1 in a million, but the problem is that these cells this reservoir does not decay with time on heart, a half-life of 44 months. Interestingly, David Margolis's group has recently repeated this analysis and come up with a half-life of 43 months, showing that all of the changes that have happened in antiretroviral therapy in the last decade have actually not helped us much with this problem of the latent reservoir. So what I'd like to talk about is how this reservoir is established. This has been a subject of great interest recently, and I think if we understand this, it may help us understand how we're going to get rid of it. This is the original hypothesis. All hypotheses about how this reservoir is established have to take into account the following observations. The latent reservoir is located in resting memory CD4 cells. Most of the viruses in this reservoir are R5 tropic. Now, interestingly, most of the studies of reservoir establishment have used X4 viruses, and this is a problem as I'll show you. R5 viruses do not readily infect resting CD4 cells. And work in the SIV system from Dan Baruch and Louis Picker has shown that reservoir establishment can be blocked by vaccine-induced CTL, but not by early antiretroviral therapy started on day 3. So to study the establishment of the reservoir, we've infected primary CD4 cells, either freshly isolated or transduced with BCL2, which allows the cells to survive in vitro for long enough for latency to be established without altering their normal physiologic responses to activating stimuli. And we've infected these cells with GFP-tagged pseudoviruses carrying either the X4 or R5 envelope. We can look at productive infection by looking at GFP expression 3 days after infection, and what you see is, as you'd expect, higher levels of infection by X4 virus and with activated CD4 cells. We can then culture the cells in the absence of activating stimuli for 4 weeks, sort the GFP negative cells, and then reactivate them and look for GFP expression as an indication of latent infection. And interestingly, when we do this with R5 viruses, we don't get any latently infected cells following infection of either activated or resting cells. Now, part of this is due to co-receptor expression. CCR5 is expressed only on a small number of resting CD4 cells. Those cells have a memory phenotype, high CD45RO, and a low-level expression of CCR7. But even if we sort those CCR5 positive cells and infect them and look for generation of latently infected cells, we don't see any. So how is this reservoir established? Well, we originally hypothesized that it was due to infection of cells that were transitioning from an activated back to a resting memory state, cells that have been termed memory precursor cells. So to study this, we activate cells for 3 days and then switch to a basal media and culture them. And if you do this, what you see is that the day 3 activated cells have high levels of CD25 and are proliferating, but very quickly thereafter, by day 6, they lose CD25 expression and exit the cell cycle. Interestingly, if you look at CCR5 expression, that continues to increase on these memory precursor cells, and is actually higher on these cells than on the activated day 3 cells, suggesting that entry would occur readily in these memory precursor cells, but other steps in the life cycle don't proceed as efficiently. To look at reverse transcription, we've infected cells at different times after activation with X4 viruses to bypass the changing levels of CCR5 expression, and then looked for late reverse transcription products 1 day after infection. And what you can see is that reverse transcription proceeds well in the activated cells, but less efficiently in cells that have been infected at further times from activation. And the same is true with virus gene expression. If you look at expression from the HIV LTR by the level of GFP on the GFP positive cells, you can see it's highest on cells infected 3 days after activation, and then declines rapidly. And this is true if you deliver the virus either by X4 infection or by a direct transfection of proviral constructs. Now, activation, of course, causes dramatic changes in the level of expression of many genes, and then after day 3, you can see sort of a relaxation back to a basal state. And interestingly, in this analysis, you can see the delayed pattern of CCR5 expression. Now, NF-kappa B is very important in driving HIV gene expression, and if we look at specifically by microarray analysis at the most upregulated NF-kappa B responsive genes on day 3, you can see that they rapidly downregulate expression. And then to look directly at the level of HIV gene expression in cells infected at different times after activation, we've sorted the GFP positive cells, individual cells, and looked at levels of transcript in single cells. And what you can see is that the level of HIV RNA is about 100-fold higher in cells infected on day 3, and then very rapidly declines, indicating that conditions for HIV gene expression rapidly become suboptimal after day 3. And so this all suggests that infection of memory precursor cells is what leads to latent infection. And so to directly test this, we've infected cells at different times after activation and then switched to basal media, cultured for 4 weeks, sorted the GFP negative cells, reactivated them, and looked for latent infection as evidenced by re-expression of GFP. So productive infection, you can see that it's highest on day 3 for X4 virus and day 6 for R5 virus. But latent infection as actually peaks on cells for R5 virus, cells infected on day 9. So cells that are transitioning back to a quiescent state. So all of this suggests that it's infection of memory precursor cells that leads to latent infection. These cells have high levels of CCR5 due to the delayed pattern of CCR5 expression, and therefore are permissive for entry. They're less permissive for reverse transcription and are rapidly losing permissiveness for HIV gene expression, allowing latency to be established. So establishment of latent infection reflects dynamic changes following activation, delayed upregulation of CCR5, and the transient nature of the optimal transcriptional environment for HIV gene expression. Interestingly, the gut-associated lymphoid tissue contains large numbers of CCR5 positive, CD4 positive T cells, and is the site of early viral replication, and therefore it's difficult to block the establishment of the latent reservoir with early antiretroviral therapy. In terms of implications for cure strategies, the reversal of latency really requires recreating the transcriptional environment of activated T cells without generating cytokine storm. No single agent may be able to do this, but agents that activate critical signaling pathways downstream of the T cell receptor may be the most effective. Agents that simply modify chromatin may not do it. And interestingly, the intimate relationship between T cell activation and HIV gene expression poses challenges for silencing strategies that Susanna is going to discuss in the next talk. Now, you can see some of these predictions playing out in early studies of the shock and kill strategy, which I'll summarize as follows. Numerous latency reversing agents have been identified in studies with transformed T cells and primary cell models. Few of them have been shown to work ex vivo in cells from patients, and in no case has there actually been a reduction in the reservoir demonstrated to date. But there is evidence for transient increases in cell-associated HIV RNA and plasma HIV RNA in studies from Dave Margolis, Sharon Lewin, and others. This is the pioneering work of Dave Margolis showing transient increases in cell-associated HIV RNA after treatment with the HDAC inhibitor vorinostat. And more recently, studies of another HDAC inhibitor have suggested actually increases in the level of plasma virus. So our own approach has been to look ex vivo in cells from patients and expose these cells to latency reversing agents and then measure cell-associated HIV RNA or virus production, always compared to a positive control, which is full T cell activation. And full T cell activation as generated by PMA and ionomycin treatment gives about a 100-fold increase in cell-associated HIV RNA. But when we've looked at agents such as the HDAC inhibitors or JQ1, we've seen really fairly modest increases in HIV RNA. The only class of agents in our hands that are really effective as sort of single drugs are PKC agonists, which activate a subset of the pathways turned on by T cell receptor activation. And interestingly, in combination with HDAC inhibitors and other agents, PKC agonists show dramatic synergy and levels of latency reversal that approach that observed with full T cell activation. If you look at virion production, it's really only the PKC agonists or combinations, including PKC agonists, that give very good levels of virus production. And interestingly, this can be done without generating cytokine storm, whereas full T cell activation generates lots of pro-inflammatory cytokines. So these results are encouraging, but reversing latency is not sufficient. We have to make sure the cells die. We're reasonably sure that reversing latency with T cell activation is going to lead to the death of infected cells, but it's not so clear with latency reversal by other approaches. We can't rely on cytolytic T cells to do the job. We've shown previously that using this BCL2 model system that latently infected cells can be cleared by CTL from elite suppressors following latency reversal. But if you look in patients on antiretroviral therapy, the CTL response is generally insufficient to clear latently infected cells following reversal of latency, unless some form of stimulation is done first. And we've recently shown that in patients who start treatment during chronic infection, virtually all of the virus in the latent reservoir has escape mutations in dominant CTL epitopes. whereas the same epitopes are 100% wild type in patients who start treatment during acute infection. And this is shown here for 3 different T cell epitopes. Now, with regard to vaccine prevention of infection, we've looked at whether or not a pre-existing CTL response can prevent the establishment of latency in the model system that I described, in which we infect memory precursor cells and then co-culture them with primed autologous cytolytic T cells. And in this case, the yield of latently affected cells is dramatically reduced, suggesting that a pre-existing CTL response might prevent establishment of the reservoir by lysing these cells before they fully enter a latent state. Now, I'd like to just conclude with a brief discussion of assays for the latent reservoir, because it's going to be critical that we be able to accurately measure the reservoir if we're going to make progress in this area. There's really 4 types of assays: the original viral outgrowth assay, simple DNA PCR assays that detect the provirus, and then assays in which cells are treated with some kind of T cell receptor agonist, and then one measures either intracellular HIV RNA or virion production. Now, in collaboration with Steve Deeks, my wife Janet recently completed a study in which we compared a variety of assays for HIV DNA with the viral outgrowth assay and found that the frequency of cells detected by DNA PCR was about 300-fold higher than and poorly correlated with the viral outgrowth assay. And this reflects the fact that there's lots of proviruses that are not induced to produce replication-competent virus in the virus outgrowth assay. Many of those have large dramatic defects, including large internal deletions or hypermutation. But a small fraction are actually intact at the primary sequence level. And if you reconstruct those viruses and ask whether they can replicate, we've shown that most of them replicate very well compared to replication-competent viruses from the same patient. So this has given us a sort of a different perspective on the size of the reservoir. This is the size of the reservoir if you do DNA PCR in different patients. This is the size if you do the viral outgrowth assay. And then this is the size if you measure the number of intact proviruses that don't have any obvious defects. So the true size of the reservoir is somewhere between the pink and blue spheres. There's about a 62-fold difference on average. And the true size really depends on how many of these intact non-induced proviruses can be induced. To address this, we've carried out repetitive stimulation experiments in which you take the cells, the wells that are negative for viral outgrowth, and simply subject them to another round of activation. And what happens is that you can induce additional viral outgrowth, as shown here. So that what it really looks like is something like this. The true size of the reservoir is something like this pink sphere. And we actually don't have a way to measure this right now. Now, recently, we've completed a more exhaustive study of the proviruses and resting CD4 cells using an unbiased screen and doing full-length single genome analysis. And this has given us really a more complete picture of what's there. In this slide, each horizontal bar is a provirus. The white areas are deletions. The light blue is hypermutation. And an intact provirus would have a dark blue line all the way across. You can see it's actually hard to find an intact provirus. In this analysis, over 98% of the proviruses detected that would be detected by DNA PCR are defective. These defects can be seen even in single-round infection. You can see both deletions and hypermutation. And even if you look in patients who start treatment during acute infection, there's lots of defective proviruses, including particularly a high fraction of ones that have hypermutation, and we estimate 91% are defective. So these defective viruses accumulate very rapidly in acute infection and greatly complicate analysis of the reservoir. So based on these sequences, we can predict what would be seen with different types of assays for the reservoir. If you simply do DNA PCR using GAG primers, this is the size you get. It's sort of a maximal estimate of reservoir size, but in fact, it misses some proviruses that have deletions in GAG. The quantitative viral outgrowth assay is really the best assay for replication-competent virus. It provides a definitive minimal estimate of reservoir size. But it doesn't pick up all of the intact proviruses which have the potential to replicate. And this number of intact proviruses is really should be the definitive maximal estimate, but unfortunately it takes lots and lots of work to define this. We know that at least some of these can replicate by sort of these repetitive stimulation experiments. And then if we look at other types of assays, so the true size of the reservoir shown in this light pink sphere. Other types of assays, for example, limiting dilutions assays in which you stimulate cells and then measure either intracellular HIV RNA or virus production, have problems. So if you measure intracellular HIV RNA, you're going to detect some defective proviruses, and you're also missing the intact proviruses that don't turn on with a single round of activation. Better is a measure of virus production, but again, you're missing viruses that don't turn on with a single round of T-cell activation. So right now we can bracket, but we cannot precisely determine the size of the latent reservoir. And I think it's very important that we understand exactly what we're measuring with these different assays, and I think that will help us. So let me just conclude by saying that we think all of the phenomenology associated with HIV persistence can be explained by a simple model in which infection of memory precursor cells leads to latent infection. Reversing latency requires recreating the transcriptional environment of activated T cells without inducing cytokine storm. Kill strategies addressing CTL exhaustion and escape mutations are going to be needed. And finally, defective viruses, proviruses accumulate rapidly in acute infection and greatly complicate measurements of the reservoir, as well as gene editing strategies that Keith is going to discuss. I want to thank people involved in this work. I've been working on this problem with my wife, Janet, for many years. Liang Shen and Kai Deng did the work on the establishment of latency. Yachi Ho did the work on non-induced proviruses. Corinne Bolin and Greg Laird did the work on latency reversing agents. Katie Brunner and Ross Pollack did the work on non-induced proviruses, and I want to thank Adam Capoferi and Daniel Zhu for help with patient recruitment and our blood donors, and thank collaborators including Steve Deeks, Doug Richmond, and funding agencies, especially AMFAR, which has been very generous in the support of this work. Thank you.